As Professor Ferrante has just told, I'm Alessandra 𝗲𝗹𝗹𝗯𝗲𝗶, and I graduated in medicinal chemistry and pharmaceutical technology at the University of Chieti-Pescara. And I'm actually enrolled as a PhD student in science and technology for sustainable development. And my industrial PhD program is financed by Italia, which is a company that deals with industrial hemp. My PhD is about cannabis and circular economy and how to enhance an agricultural supply chain with a high social impact for the development of agro-industry through botanical innovations and related applications in the nutraceutical, pharmacological, cosmetic and cosmeceutic, and in general healthcare fields. So during my research work in the university laboratory, we have carried out different type of extractions and from different type of matrix. And we have done HPLC analysis, and now I'm developing a method for quantify and identify a huge number of polyphenolic compounds in different extracts. While in the laboratory of Italia, I'm working as a quality controller in the research and development unit for cannabinoids, lands, and irrigation waters, in order to improve the botanical cultivation of the hemp, industrial hemp. So, firstly, I just want to talk about bibliographic research, something that is an important preliminary study, because thanks to the bibliographic research, we can focus on the main topic, in this case, the plant, and the application fields, for example, the socioeconomic, legal framework, and the chemicals. For example, during my research work, I've done bibliographic research to know what are the most important chemicals in the industrial hemp, what is the range between we can use these these chemicals, so the legal framework, and how it is applied in the social sphere. After that, we have to improve the extraction. So after improved the information about the plant, the chemicals, we can go on with the extractions. That is a separation process consisting of the separation of compounds from a matrix. The analyte moves from the initial solvent into the extracting solvent according to the partition theory. There are different types of extractions. We can have liquid-liquid, solid phase, supercritical fluid extraction, ultrasound-assisted extraction, and microwave-assisted extractions, and others. There are different parameters that need to be considered for the extractions. For example, time, temperature, plant material, solvent, and polarity of chemicals. In fact, if we want to extract polar chemicals, we should use a polar solvent. And if we want to extract less polar chemicals, for example, terpene or terpenophenolics, we should use a less polar solvent like methanol or acetonitrile. Chromatography is a biophysical technique that enables the separation and the identification and purification of the components of a mixture for qualitative and quantitative analysis. Here, the molecules it is very difficult to explain. Molecules in a mixture are separated according to their chemical affinity between two phases that are the stationary phase, that is fixed and can be a solid or a liquid properly supported, and the mobile phase, that is a liquid or a gas that flows over the fixed phases. Between these two phases, there can be interaction molecules, and these interactions can be weak. For example, hydrogen bonds, dipole-dipole interactions, van der Waals bonds, and steric interactions. But there can be even coordination compounds and ion exchange mechanism. So we can classify the chromatography with the separation mechanism by the type of mobile phase and by the elution technique. Okay, surfing the separation mechanism, we are surface adsorption, and on the surface of the stationary phase, there are active sites that can establish reversible secondary bonds with the molecules of the mixture. Separation is due to a series of adsorption-desorption steps. In the partition chromatography, compounds are divided between the two phases, immiscible to each other, according to their different solubility in each of them. Here we can have two types of chromatography technique, the normal phase and the reversed phase. In the normal phase, the stationary phase is polar and the mobile phase is non-polar, while in the reversed phase, we have non-polar stationary phase with the polar mobile phase. In the ion exchange chromatography, electrostatic interactions between charge groups and solid support materials appears. Separation, so, is based on exchange of ions between surface and eluents. While in the gel permeation, the stationary phase is an inert solid, like a gel with pores, and the molecules to be separated are dissolved in the mobile phase. The separation occurs according to the difference in molecular size. The type of mobile phase is another way to classify chromatography, and we can have liquid chromatography, where the mobile phase is a liquid where the sample ions or molecules are dissolved. Gas chromatography, where the mobile phase is a gas in which compounds can be vaporized without decomposition. And the supercritical fluid chromatography, where the mobile phase is a supercritical fluid like carbon dioxide. The elution technique can be different, so we can have planar chromatography, where the stationary phase of the process takes place on a plane. So we can have paper chromatography and thin layer chromatography. While in the column chromatography, compounds move through a column. Chromatogram is a detector signal that describes the progression of the analysis and is in function of the time of the or of the volume of the eluent. High performance liquid chromatography or HPLC is a technique, analytical technique to separate, identify, and quantify each component in a mixture. It's characterized by high pressure inlet, about 50 to 350 bar, and due to the small amount of samples, we have column with 2.1 and 4.6 millimeter diameters and 30 to 250 millimeter length, while the stationary phase is constituted by very little particles, 3 and 10 micrometers. Those characteristics give a superior resolving power to the HPLC and to the purpose of the HPLC that begin a popular chromatographic technique. Important is to remember that HPLC is distinguished from traditional low pressure liquid chromatography because of the operational pressure that are significantly higher, while in the ordinary liquid chromatography, the force that flows the mobile phase into the column is only the force of gravity. The advantages of these chromatographic techniques are different, and we have fast analysis, high resolution, high sensitivity, recovery of samples, and use for a huge number of molecules. HPLC instrumentation is schematically consisted in mobile phases, a degasser that removes air bubbles from mobile phases, pumps that deliver flow and composition of mobile phases. And we have the mixer that can mix the different solvent for mobile phase, sampler that brings a sample mixture into mobile phase, injector. Sometimes we can have even a pre-column, a column, analytical column, and then we have the detector that generates signals proportionally to amount sample, and then software for data analysis. The mobile phase should have different characteristics to be used. For example, high purity, low cost, UV detectable, non-corrosive, low viscosity, low toxicity, non-inflammable, and high selectivity for selected compounds. Some examples of mobile phases can be acetonitrile, methanol, water, hexane, and toluene. We can even use modifiers for the mobile phase, for example, buffer solution that can be used for pH adjustment. And if we are working on a reversed phase chromatography, we can use acidifiers to suppress the ionization of acid analytes and alkaline modifiers to suppress the ionization of alkaline analytes. Pumps provide a high inlet pressure, as we were talking before. And it is important that even the flow is pulsation-free. Then inside the pumps, the wettable components are made of inert material, and some models of pumps can mix multiple solvents together in ratio, changing in time, generating a composition gradient in the mobile phase. Injector can be manual or automated, so we can have the autosampler. And the injection volume and technique are exactly the same for each injection. The amount of sample injection is in a range between 1 to 100 microliters, and this gives the HPLC a high degree of injection volume precision. For columns, sometimes we can use, it is recommended to use a guard column, pre-column, that is 2 centimeter length and 4.6 millimeter diameter. Then we have the filled, we are talking about HPLC, that is a kind of chromatography. So there are different types of sectionary phase. And so columns are filled with adsorbents varying in particle size, porosity, and surface chemistry. Smaller particle size are packing materials requires the use of higher working pressures and typically improves chromatographic resolution. Particle diameters between 3 and 10 micrometers, as we were talking before. The most common used for columns is a silica, and the temperature column oven can be set for better separation. So talking about normal phase chromatography, we have said that we have polar sectionary phase with the non-polar mobile phase, where the R groups of the sectionary phase can be diol, amino, cyano, inorganic oxides, or dimethylamino. And in the normal phase chromatography, non-polar compounds will elute first, while in the reversed phase chromatography, non-polar sectionary phase is added with the polar mobile phase. R groups are of the sectionary phase are hydrocarbon, such as C8 or C18, and we can use water as a mobile phase, and that is very important. The most polar compounds will elute first. Even the elution mode is important, and can be of different type. We can have isocratic elution mode, where the mobile phase composition remains constant through the procedure, and the gradient elution mode, where the mobile phase composition changes during the separation. The most common used HPLC detectors measure a solute property by responding to the physical or chemical property of the solute. Most used detectors are UV or photodiode array. UV detector generally only measures a specific wavelength, while in the photodiode array detector, we can have a range of wavelength in real time. The HPLC has many applications in chemicals and clinical sciences, and it is a common technique used for quality control, analysis of natural contamination in water, sea, oil, and others. For clinical tests is used too, and for pharmaceutical development, for example, for drug stability or for quantification of drugs in pharmaceutical dosage forms, and obviously it is used for research purpose. So cannabis is a plant of Cannabaceae family, and we have Cannabis sativa, Cannabis indica, and Cannabis ruderalis. Cannabis is characterized by terpenophenolic compounds like THC or D9-tetrahydrocannabinol, that is a psychoactive, and cannabidiol, cannabigerol, cannabichromane, that are non-psychoactive. The action of these terpenophenolic compounds is expressed through the binding to cannabinoid receptors. And we have two kinds of cannabinoid receptors, CB1 and CB2. CB1 is located at presynaptic level, and the stimulation effects include antiemetic, antioxidant, hypotensive, immunosuppressive, anti-inflammatory, analgesic, antispasmodic, and euphoric effects. The most important cannabinoids that bind this type of receptor is the THC, so the psychoactive cannabinoid, while the CB2 receptors are located in at peripheric level, and the stimulation effects include anti-inflammatory and immunomodulatory effects. Cannabinoids for these receptors are cannabidiol, cannabigerol, and cannabichromane. The content of THC must be lower than 0.2 in Europe, and the interest for this crop has conducted to the development of new varieties that have been certified for the low content of tetrahydrocannabinol. So chemical composition, biological activity of aqueous fraction has been so far poorly investigated. And this is why we are going to talk about phytochemical and pharmacological investigations of the water extract from female inflorescences of the industrial hemp Futura 65. In the industrial hemp chain production is important now the valorization of the use of the female inflorescence that are actually less used because are considered as waste product in the fiber production chain. Female inflorescences are even sources of bioactive extracts and biomolecules of pharmaceutical interest. The aim of this study was to investigate the phytochemical composition and skin protective effects induced by the water extract from the female inflorescences of the certified Futura 65 variety industrial hemp. Female inflorescences from cultivar Futura 65 have been manually harvested from plants at full blooming state. The plants were cultivated in Abruzzo region, and the plant were dried in a ventilated oven at 40 degrees Celsius and weighted and homogenized with the extraction solvent, in this case water. And then ultrasound assisted extraction was carried out. In order to unravel the phytochemical composition of the water extract, we conducted an Hplc-Ms analysis that shows that vanillic acid, ferulic acid, and rutin and naringenin as the prominent phytocompounds in the water extract. The chromatographic run was about 30 minutes and was conducted in a gradient illusion mode with a mobile phase constituted by 0.1 of formic acid in water and methanol. While for the terpenophenolic compound, we used the HPLC coupled to UV detector and the mass spectrometer, and the phytochemical composition was constituted by cannabidiolic acid and the cannabidiol as the prominent terpenophenols in contrast with the others terpenophenolic compounds. So the phytochemical composition of the extract agrees with the antioxidant, anti-inflammatory, and antimicrobial activities of the water extract of Futura-65. So in the vitro model, for the in vitro model, we have carried out an MTT assay with the purpose of assessing the cytoprotective and antioxidant anti-inflammatory effects of the water extract. We have carried out the experiment in the human keratinocytes and the primary human gingival fibroblast. Cells were first challenged with hydrogen peroxide, and the viability was decreased. While after the treatment with the Futura-65, the viability, the number of apoptotic cells decrease almost in all conditions. Keratinocytes are involved in skin inflammatory response, releasing pro-inflammatory mediators, for example, interleukin-6, that is a key modulator of the inflammatory and the reparative process in skin. Here we have used the administration of hydrogen peroxide, and the interleukin-6 production was decreased in accordance with the reduced gene expression. After the administration of Futura-65, was partially restored the interleukin-6 levels. For the ex vivo models, we used the isolated skin tissues challenged with the hydrogen peroxide, inducing oxidative stress and the tyrosinase activity. Then the tissues were stimulated with the water extract of Futura-65. And the water extract reduced hydrogen peroxide-induced levodopa turnover, prostaglandin E2 production, and the ratio kynurenine-tryptophan, thus corroborating anti-inflammatory and antioxidant effects. The conclusion of the water extracts are that the present findings highlight the potential of water extract from industrial inflorescence as a skin protective agent. This study was supported by the publishing of on the journal Antioxidants. Female inflorescences, essential oils, have been described as effective insecticide, antibacterial, and antiproliferative agents. So we have conducted a phytochemical investigation and pharmacological for the pharmacological applications of the essential oils from female inflorescences of 3 industrial varieties. The 3 selected cultivars were Eletta campana, Carmagnola selezionata, and Futura-65. The 3 selected cultivars were cultivated in Abruzzo region in Italy, and the sowing was performed between mid and late March 2019, with no fertilization and only 3 programmed automated irrigation cycles. Manual collection of the only mature flowers during the flowering was done. Then the plant material was immediately transferred to the laboratory and extracted within 6 hours. Samples of essential oils for analytical characterization and biological tests were dehydrated through a passage in anhydrous sodium sulfate and storage in a dark glass bottle with a PTFE septa cap at 4 degrees Celsius until used. In order to unravel the phytochemical composition of the essential oils, we characterized the terpene profile of the essential oils using a gas chromatography coupled to a mass spectrometer. And 𝗕𝗲𝗹𝗹𝗯𝗲𝗶 and alpha-pinene are the most representative terpenes in all tested samples. while essential oils were analyzed for terpenophenolic quantitative determination and using a reversed phase HPLC-coupled-2 UV detector and a mass spectrometer, where CBDA or cannabidiolic acid was the most important terpenophenol in all the essential oils, and especially in the range of 57 to 70%. In the present study, we have tested the antimycotic properties towards several dermatophyte species. The essential oils were effective in inhibiting the growth of all tested dermatophytes. Essential oils, in fact, are more potent than the reference antimycotic drug griseofulvin, and Futura-65 variety was the higher with the higher mycostatic activity compared to the other two cultivars. In view of the potential applications as skin protective agents, we evaluated the inhibitory effects of the essential oils against the activity of tyrosinase, that is an enzyme that plays a key role in melanogenesis. So Eletta campana was the most active as enzyme inhibitory agents, maybe because of his higher scavenging and reducing activity. In parallel, we evaluated the inhibitory effects of the essential oils against the production of the prostaglandin E2 in an ex vivo toxicity model, incubating the isolated skin tissues challenged with hydrogen peroxide and inducing oxidative stress. Then we have stimulated the tissues with the essential oils, and the inhibitory effect induced by essential oils on hydrogen peroxide induced the PGE2 level in isolated mouse skin are shown in the figure. Hemp essential oils were also investigated in an in vitro model to study effects on cell viability. The cell line used was the human 𝗕_1299 lung adenocarcinoma cells, and that were treated with the different concentration of essential oils for 24 hours. The essential oils did not alter cell number, excluding any significant effect on cell survival in basal condition. But the capability of the essential oils to reduce the cell viability could be related, albeit partially, to the tumor cell line sensitivity. The null effect on cell viability is also mirrored by essential oils induced stimulation, simultaneous inhibition. The null effect on cell viability is also mirrored by essential oils induced stimulation. Sorry, I'm a little bit need the water. So the null effect on cell viability is also mirrored by hemp essential oils induced the simultaneous inhibition of the gene expression of TGF-beta and BCL2, which are known to regulate apoptosis in opposite ways. Regarding to the essential oils of Futura-65, PCR real-time showed that a significant inhibition of BCL2 and TGF-beta at all tested concentrations in the human 𝗕_1299 lung adenocarcinoma cells. Whereas the essential oils of the two cultivars did not significantly alter gene expression. Also considering the recent interest of the scientific research toward the hemp-deriving extract as putative anti-COVID-19 agents, we investigated the influence of the essential oils on gene expression of angiotensin converting enzyme 2 and the transmembrane protease serine 2, which besides being co-expressed, are reputed to play master roles in mediating SARS-CoV-2 virus entry in the human host. The essential oils were assayed for evaluating their influence on the ACE2 and TMPRSS2 gene expression as well. As depicted in the figures, all essential oils were able to significantly inhibit the gene expression of both ACA2 and the TMPRSS2 in H1299 cells, suggesting their possible use in protective devices for counteracting the SARS-CoV-2 virus entry in the human host. Concluding, the present findings highlight the rationale for improving the whole botanical chain of the industrial hemp, considering the inflorescence byproduct as a valuable source of biomolecules with pharmacological applications. And specifically, the present study aims to be a starting point for the development of innovative topical pharmaceutical preparations and devices for counteracting the inflammatory burden of oxidative stress and inflammation occurring during dermatophytes infections. This work was supported by the publication on antibiotics, and I've done. So thank you for your attention. Thank you, everybody. And that's all.